Thank you for having me. And I don't think I could have asked for a better lead-in than the previous talk or the previous question, as I'm going to be talking about how we think about pancreatic cancer in the spatial biology context. So let's see if I can figure out this computer. Is this? Yeah, it'd be great. Let's see. How do I? Oh, okay. So can you advance with this? Sorry. Okay, there we go. So go back. So just briefly, my disclosures now that I've figured out the technology here. So, the way I sort of start to think about pancreatic cancer is we're doing a lot of work in thinking about early detection of pancreatic cancer and understanding the mechanisms by which it gets there. And one of the reasons this is so critical is that unfortunately for most patients, by the time they get diagnosed, it's too late for most people. It's at very late stage. The tumor has a very dense immunosuppressive microenvironment and is basically universally resistant to almost any therapeutic that's given to it because it's silent. And from a spatial biology perspective, it also poses a lot of interesting challenges because in contrast to many other tumors, when you look at a pancreatic tumor, only about 10% of the cells are actually tumor cells. The vast majority of the tumor is macrophages, fibroblasts, and other components of the microenvironment. So it makes it really critical as we start to understand this tumor to take a spatial biology lens in order to figure out what's in there and what's going on and how all these different cell types interact. So while we're at a spatial biology meeting, I'm going to start with some of the early work that we did in single cell biology in order to start teasing apart the different cell populations that were there to start understanding some of the communication processes within the late stage immunosuppressive environment. So not just because it was the pandemic, we, like everybody, collated a public domain atlas of the data that were there as a training exercise. And the inevitable thing you do when you get single cell or initial spatial cohorts, right, is you run clustering analysis, you figure out what cell types were there from a bunch of these. And within these cohorts in single cell, we were able to identify, yes, tumor cells, myeloid cells, CAFs, interestingly, in a smaller proportion in single cell technologies because so many of those go away in the dissociation process, but see broadly what cell types were there. Of course, if all we're going to do is go cluster the data and infer what cell types are there in the population, we probably wouldn't need this broader meeting because there are a lot cheaper and easier technologies to go through and start characterizing what cell types are present in the tumor. So one of the things that I'd like to emphasize is that when we have these high dimensional assays that we're all using, some of the power of them is that we can go beyond what cell types are there. And as Wagner and all beautifully summarized in their review, say, okay, yes, there are cell types that are identifiers, but in addition, the high dimensional information has footprints of where's the cell phenotypically, where is it temporally in terms of evolution, where is it in cell cycle, where is it spatially. All of these leave different signatures on the data that it's our job as bioinformaticians to figure out what's going on and how do all these different signatures come together in order to learn what's going on within the microenvironment of these tumors. So, sorry, this is a very sensitive pointer, clearly. So Genevieve Stein O'Brien, who was a postdoc in my group at the time that single cell technologies were starting to emerge, realized that these axes of identity that you have of different cellular signatures perfectly maps to the mathematics of matrix factorization. So I'm going to take a step back and explain a little bit what I mean about that. So if we imagine instead of measuring 20,000 genes, we are now in a cartoon world that I've drawn here with only 4 genes, whose expression is drawn on the y-axis. And in the x-axis, I show different cells. And you can imagine the left being tumor, the right being normal, for example. We can very clearly by eye see that there's 2 distinct patterns in the data, one of which is associated with the left half with the tumor, one of which is associated with the right half. And every single gene is using either one or the other or some combination thereof. And what matrix factorization lets you do is learn these, what we call patterns in the data, right? Learn what are the features that are being used in different cellular groups, and then assign gene weights to them to say, what are the genes that are being associated with each pattern? So we've used a particular method in our group that we've developed called COGAPS, which is a non-negative matrix factorization. And we like it because we view this additive process of adding up the things that are there as more reflecting what a cell does. The cell doesn't necessarily, yes, it can delete a gene, but biological processes by and large tend to be more additive in the population. So what we sought to do is say, okay, can we look at, let's just start with the tumor cells. And can we see what types of transitions that we observe as a cell goes from being normal ductal to tumor and to a tumor? And how does that change if it's a tumor adjacent normal cell relative to a true normal cell? So we took this population of tumor cells and work led by Jacob Mitchell and Jeanette Johnson, who's sitting in the back of the room here, and started to say, okay, can we apply this matrix factorization approach in order to understand these transitions? So when we applied our approach to the data, what we observed were two predominant patterns in the data, one of which was not terribly shocking. We saw a subset of the tumor cells were associated with KRAS signaling and estrogen response signaling, and a number of pathways that were largely associated with cellular proliferation. The other signature was also not terribly shocking. We saw inflammatory response and epithelial to mesenchymal transition that was mutually exclusive with cell proliferation in another set of tumor cells. What surprised us a little bit more was that we observed the same EMT and inflammatory signal program also occur within the tumor adjacent normal cells, leading to us to hypothesize that there must be something going on in the transition that occurs when you're with a tumor adjacent normal cell relative to being a true normal ductal cell within the pancreas that might be stressing it and causing that alternative response. So the advantage of the single cell technology, right, is that we have not only, what cells are there, but not only these patterns, but we have all the other cells that are there in the microenvironment. So we went back to our cohorts of public domain data and correlated these patterns with what we saw of the density of all the different cells in the environment. And we observed that at least in one of our two cohorts, this pattern was predominantly occurring, and it was correlated with the density of cancer-associated fibroblasts within that sample, which led us to hypothesize that it was probably cancer-associated fibroblasts that are inducing this inflammatory response and this EMT-like transition. But of course, this only occurred in one of the two data sets that were there in our cohort, which as a computational biologist makes me immediately think I've done something wrong and there's some technical artifact in my data. So the question is, if this is all the data that we have, how do we start looking at and using experimental systems to validate that is this just some artifact, or is this actually something that occurs within biological systems when you have fibroblasts interacting with epithelial cells? So I was fortunate that there was a graduate student, Samantha Gwynn, who was in the labs of Jackie Zimmerman and Rick Burkhart. And she had developed a very elegant organoid co-culture model of calves and organoids that we were able to then look in isolation. What happens if you have just the tumor cells, just the epithelial cells? What happens if you have the fibroblasts? And then what happens if you bring them together? And I realized the p-values did not convert from Mac to PC on the screen, but I assure you they are statistically significant if we convert in software. That what we saw is that if we looked at single cell data in one of these organoids, we saw indeed an enrichment of this pattern in the co-culture relative to monoculture. And what was even more interesting was when we looked across a whole panel of organoids and ran bulk RNA-seq on these and looked at the projection, we saw it also. And this EMT signaling was increasing independent of the subtype of pancreatic cancer. So the CAFs, we don't see a subtype shift, but we do see a shift in this state towards more EMT-like, leading us to hypothesize that this is more of a phenotypic plasticity relative to a real subtype difference, as has been commonly denoted in the classical and basal subtypes of pancreatic cancer. So that led us to the end of our cancer research paper, where we have a nice little cartoon diagram where we wrap it up in a bow and pretend we know exactly what's going on, where now we say we have normal cells that we hypothesize are transitioning to more of a mesenchymal-like state through contact with the fibroblasts and cell signaling, that the same type of process occurs within the tumor cells, but in the context of the tumor cells, this process we think is mutually exclusive with cellular proliferation. So as in Sam's follow-up work, typically this would lead to, the next experiment to look at the molecular mechanism, the new therapeutic to overcome resistance. But, these are both really not accessible as much to my lab as a computational biologist. And so we want to see, if we know what these are, can we start to understand what are the systemic implications of the inferred mechanisms? And can we start to use the computer to conduct experiments entirely in silico before we ever go back to a murine system or going back to certainly a human clinical trial? So the way I like to think about it is that when we're thinking about spatial biology or single cell biology, a lot of what we think of computational biology as being is very data-driven, very transactional. Somebody has a data set, and you use a computational method in order to infer what's going on within that data set and learning the types of signals that are in that data set. In contrast, there's a whole other field of computational biology of mechanistic mathematical models, which say, if I know what cells or molecules do, can I build up a system of equations from first principles and use those to probe the system? So the analogy I like to give for this, and being trained on this very campus, in the applied math group at the interface with several physicists, is that if you were throwing a ball up in the air and trying to figure out where it was going to fall and what the implications of that, it would be highly inefficient to measure every single point along the trajectory of a ball, fit a parabola to it, and use that to figure out where the ball is going. And I would argue that's a lot of what we're doing in this type of characterization work. What you would do instead is go, okay, I know the force of gravity. I generally know how that works. So I know all I need to know is the initial condition of how fast am I throwing the ball, what direction, and what's the height. And then I can use a series of equations based on the force of gravity to figure out where it's going to go forward in time. So we want to start adapting those principles now that we're learning more of the rules of cell-cell interactions. Can we adapt those same types of principles to biological systems to start predicting where biology is going to go, and what are the implications of these wiring diagrams that we're learning from all of our analyses? So this requires that we start turning our biological systems into word problems, which, I realize is not necessarily everybody's favorite cup of tea, though I always enjoy it. And so starting to think about, as we're using these data sets, can we use them to figure out what variables are we tracking, sort of what's most critical to the system, what factors, what cell-cell interactions, or what intracellular mechanisms within the cell are regulating that. And then we need to know what are the equations that regulate their evolution. So this historically has been a very labor-intensive process, and part of why this hasn't been done as much is because each new set of equations requires custom code to adopt. So I had a grad student coming in to start getting fibroblast biology, and that was his entire PhD project, was building out the equations to extend the model from fibroblasts. So it doesn't really scale very well in most contexts. We were very lucky to start a collaboration with Paul Macklin, who's developed an agent-based modeling software called PhysiCell that does enable this to go a lot more at scale. And the idea behind this system is, let's say we have a series of cells. We know what they're called and we know what they do. And then we know how they interact with other cells and biophysical properties in their environment. Then he's developed a series of software that says, I will evaluate forward in time at each step based on where cells are in their environment, what this is going to do using what's called an agent-based modeling framework. So again, this also was very cumbersome to work with, required sort of coding and a lot of XMLs and things like that, and made it added an extra step of gatekeeping away from the single cell and spatial biology assays that all of us are using in terms of how do we think of bridging the real world data with these types of simulations to predict the implications. And so what Paul did, and work led by Daniel and Jeanette, again, back of the room, was to say, can we translate this software for agent-based models into plain language? So if we have all these equations that we've encoded within our software base for these types of things, largely there's a series of set common behaviors that cells are doing. There's a dictionary of stimuli, what talks to a cell, what causes it to change behavior. There's a dictionary of behaviors. What are the things that cells do that are categorized? And then there's a grammar that connects the signals to the behavioral response. So the idea was, could we translate this, instead of having to use the intense software code for this, into plain words languages? For example, CAF induces EMT in epithelial cell, and have it parse behind the scenes and convert that sentence into an equation. And so we started exactly with our case of the CAF PDAC example that I was talking about. And exactly like I was saying, now we're able to write this whole system that we had inferred in a network diagram in plain language, as you can see here. And these sentences become our entire mathematical model. So instead of having to write out a series of equations, which can be a little bit cumbersome for people, we hope that this, series of sentences that are relatively English in terms of biology are going to help bridge the mathematical modeling and the biological and bioinformatics disciplines so that we can start to work with these and automatically generate. So I'm going to now show an example of what this looks like in a model that Daniel initially wrote, where we have the fibroblasts in yellow that are around the tumor, the normal epithelial cells in gray, and then they transition to mesenchymal based on their interactions, which we're hypothesizing is the transition mechanism into the tumor. So what you can see is that the cells start to transition when they interact with fibroblasts after some time. The EMT-like cells are moving a little bit more. They're able to go out and get away from the fibroblasts and escape. And then eventually what happens is that when the cells get far enough away, then they're able to colonize and start seeding out tumors. What's interesting about this observation in this simulation is one of the big puzzles that's been there in pancreatic cancer biology is that what's thought is that the basal-like phenotype, which is associated with more EMT, is commonly associated with metastasis. But if you look at the invasive lesions transcriptionally or the metastases, they switch subtype back to classical, back to being more proliferative. And we think just this simple mathematical model of the role of EMT induced by CAFs would explain exactly why that would happen without needing any more complicated mechanisms in the system. The other nice thing about in silico models with this is now that we have this here, I can test the impact of different CAF densities on the role of invasion in as many experiments as I want, right? Because it's just the computer running these. Sorry. In principle, if I can get the mouse to play right on this thing. So hopefully these will all play. Yeah, if you can play all three, it's not letting me do that. So what you'll see as these play forward is that when you don't have a lot of CAFs, as is the example on the right, the tumor just expands, becomes all basal. And you have a couple of regions where it starts to probe out, but not that many. Whereas if you have a lot of dense fibroblasts, it stays a little bit smaller and more constrained, but you start to see more invasive lesions coming out. Again,
properties in PDAP and are necessary components of PDAP information. So this is a spatial biology meeting, so I probably should be talking about some spatial data. The other nice thing is that once we found these gene signatures, we can project them.
Visium spatial transcriptomics data. Is it not showing? Okay. We're like 3 slides ahead. Sorry. So we can project up. This is way. Can I go forward one, please? Okay. So we're able to then project the data on the same gene expression signatures that we observe from single cell data onto the spatial transcriptomics data. And we can see the same mutual exclusivity between the EMT-like signaling and the proliferative signaling in different regions of the sample across our whole regions. So now, if we actually know the spatial distribution, we wanted to see, could we use that spatial distribution that we know from real-world spatial transcriptomics data of tumors in order to build out and initialize our mathematical models onto tissue? So Daniel developed the bioinformatics walkthrough. And the idea behind this walkthrough is, let's say you have your single cell or spatial data set that you've analyzed, and you've got it down to a list of cell types that you've inferred. Now, what that software will let you do is read out the labels of your single cell object in the metadata and automatically pull down what cell types are there so that you can start to form the grammar for the types of rules that you would want to encode based on the cells that are there. It then says, okay, I know what the different distributions of the cells are. And in the case of spatial transcriptomics, I can initialize the cells instead of randomly, like I was showing you, where I see them spatially. And if it's single cell, I can initialize them in the right density, but in different types of spatial distributions, and then build our mechanistic model. So we applied this to 2 different PDAC tumors, which hopefully both movies will play. So we have 2 different PDAC tumors in this case. You can see the transition to more EMT-like gradually, where it appears to go from more classical to more basal. And then again, as we get these invasive lesions forming, we see the transition back to a more epithelial-like classical state in the invasive front. So the next thing that's also exciting about this that you can't do as easily in real biology, perhaps with CRISPR, but other mechanisms, but it's harder, is we can also modify the rules that went in. And we can start to do experiments computationally on our very hypotheses of the mechanisms of cell-cell interaction. And so what we had observed when we had gone through the bioinformatics analysis, that it appeared that the signaling between the epithelial cells and the fibroblasts was occurring through collagen, and therefore we were hypothesizing regulated by the ECM. And that was all the rules that we built into our model. Alternatively, this could have happened from direct cell-cell contact between epithelial cells and fibroblasts. So what Jeanette next did was to say, okay, if I take this model and I change the rules so that it's relying on direct cell-cell contact as opposed to the ECM modulation, what happens? In that case, what we observed was absolutely explosive tumor growth almost immediately, with these little mesenchymal bits around the edges and more of an epithelial core in the tumor. And we lost the transition through the basal-like state before invasion formed. So it seemed a lot less realistic. So this again led us to hypothesize that it must be some sort of a durable secreted mechanism that was occurring. So again, how do we go back and ensure ourselves that this isn't just a bunch of computational artifacts and actually reflect some biological reality? Then what we were able to do is say, okay, based on this, we hypothesized that calf conditioned media would have the same effect on invasion that we're observing in our samples. So Laura Wood was kind enough to perform that experiment for us. And indeed, what we observed is that when we grew the epithelial cells in calf conditioned media, whether they were the iCalf subtype or the myCalf subtype didn't matter, we started to see this more invasive progression in PDAC regardless of that. So leading, again, sort of supporting the idea that there's some ECM-based modulation occurring from this. So I'm gonna skip past this. Sorry, in interest of time. But suffice it to say, we also have now, there's an angle that you can use of this to then also start simulating therapeutics. So with that, I hope that, one of the things that we really would like to think about is going from pure data driven to starting to think, can we use these virtual tools for predictive modeling of cancer? And we like to think of them as means of refining our hypotheses. If we have a hypothesis about how cells are interacting, it can oftentimes be too complicated in our head to figure out all the interactions between cells. Can we use this framework to ask if-then questions to test what our expectations are and what are the implications of this to start refining the types of experiments that we're doing? And I'd like to thank all the members of the lab, and amazing team of collaborators who were willing to bear with us on this crazy mission, for their work, as well as funders, and happy to take questions.
Any questions? All right, in the back.
Wonderful talk. Thank you so much for that. I was just curious to know how the sensitivity or the scalability of the model would be if we had to introduce a lot more variables at different scales, different levels. If you could talk a little more. Thank you.
Yeah, so I think at some point, if you have too many variables, so there's two components, right? There's what's the sensitivity of the model to the parameters that we used. So we found in the epithelial calf case, we didn't see a huge amount of sensitivity order of magnitude to those. And I'm pretty sure based on the experimental data that getting to the resolution of iCalf, like different calf subtypes, I don't think that would have been terribly relevant to the results. What we did find is that immune cells that are moving a little bit faster, there the model's a lot more sensitive to the rate of parameters that are occurring. In terms of number of cells that you want to add, I think you have to be careful. That I think I've noticed a tendency of a lot of collaborators want to try to add everything to the system. And the problem is, as you do that, it's a lot more computationally inefficient. You lose some of the interpretability, I would argue, to isolate individual components. And you also end up with so many parameters. It can be really difficult to test them and estimate them. So I like to think about this as sort of like the computational version of organoid co-cultures, right? You would never try to throw everything in your organoid all at once. I guess there are some systems that do that, but typically you're going to try to use this to tease apart individual behaviors. There's certainly work to think about how we could make this a full-on avatar, but we're nowhere near there yet.
Beautiful talk. And my question is, you're applying the spatial transcriptomics, which is really exciting. Is there any way to, now that, like, phrasing the question is like, so many people now you see these studies taking H&E and then imputing, and so I guess what I'm wondering is, if you forecast the future that you'd be able to take an H&E or a low plex that's in the clinical setting and then kind of infer some of these fibroblasts and train your model basically using a very clinical setting. You know what I'm trying to ask?
Yeah. And I will say I buried it under the rug, but we actually used the H&E from the spatial transcriptomics to estimate the collagen alignment and ECM that initialized our model. So there was part of that already. We're working with Ashley Keeman and Youngwang Chung, both of whom are developing some of these AI-based estimations for H&E. And a lot of what we're doing with this is saying, we're using the AI right now when we're thinking about the spatial biology part, is where do you have sort of the most bizarre cellular regions that you don't, either you can't resolve the morphology or that it's a really interesting phenotype, for example, like right at the invasive front, and using that to figure out and only do the spatial transcriptomics there and trying to impute back on the H&E. From what we've been seeing in our gene signatures from the matrix factorization, if we look at the epithelial cells, we basically, and I've run, across every type of precursor lesion in pancreas cancer at this point, we basically see they proliferate, they EMT, or they have inflammatory signaling. And that's most of what we see. And at that point, I think you can infer a lot of that from AI on the H&E. I'm not sure you need spatial transcriptomics for all of it. So I hope that answers the question. And the software that Daniel wrote for the walkthrough is totally agnostic to data platform. It's available for any spatial assay. So the one trick is that it assumes cells are all spherical and equal sizes right now. So you have to make some scaling for that, but it can work across any imaging technology.
All right. Thank you very much. That's Fertig. If there's no more questions.
Thank you.
Oh, there's one. Last one.
Hey, Elana. Great talk. I was just wondering, is there no like core program which proximity to the fibroblasts doesn't seem to affect? Like, at what point does proximity become secondary?
It's a good question. I don't know if anybody knows that. We've, in some of the spatial work, we've been showing that if you have regions, sort of isolated regions without fibroblasts, you can get immune cells coming in. And so I don't know if you were to look at it from a cellular sort of ecosystem level, because if you have sort of just a little bit of a break in that region and then the immune cells coming in, that might drive it more.
Okay.
But I think it sort of depends phenotypically on what's happening in the epithelial cells. In PDAC, what you tend to see is almost all the samples, you have these epithelial cells and they're almost universally surrounded by fibroblasts. So it's very rare that you see samples that don't have that in this context. But I think it would be in other diseases, I think that's probably more common.